Okay, welcome back, everyone. This is our fourth lecture. Hard to believe the semester is moving as fast as it is. We're going to talk about some of the main mechanisms that you run into in these kinds of simulations today. And we'll try to do some simulations in class involving cell sorting. And I hope, depending on how the class goes, we'll get to talk about some cell growth and division problems. Some of these things are going to be things that you talked about a little bit or tried out a little bit in the homework that will go over some of those issues. To be maximally efficient, so I'll review what we did last time. There are a couple of small issues that I'd like to cover, like how to reference cell types by name, do the homework. I'm going to move the discussion of project ideas to the very beginning so that the people who are involved in that, who aren't otherwise needing to be on the lecture, can do that. And what I'd like, again, is a very, very short summary from each person. Certainly no more than 5 minutes of where you are with your project ideas. I want by the end of this week, I think it's essential that everybody be teamed up with somebody, whether that's Josh, TJ, me, Giuliano, to help out on the project and have a basic idea of what the project is going to be. I know several of you have made some good progress, and then we should be scheduling basically 2 hours a week, one-on-ones for each person with whoever they're working with, to try to get these projects delivered. Then we're going to talk a little bit about how to specify cell volumes and surface area. That was covered a little bit in the homework, so it'll be a bit of a review. Idea of pressure and surface tension, also covered a little bit in the homework, and so review. And then we'll do a few other things. Tracking fields was in the homework, see if people were able to get that. And then we'll talk about contact energies of cell adhesion. It's one of the most important mechanisms that you run into in biology. As my friend Gabor Forgax and Stuart Newman's textbook says, the very first sentence of their textbook on the physics of multicellularity says, most important things cells do are stick to each other, because otherwise we'd be unicellular organisms. A funny joke, but also true. And we'll work around with cell adhesion a little bit. And then I hope we'll be able to do a little bit on cell growth and division. And with that, you'll actually know most of the main mechanisms in CompuCell. What we won't have talked about are chemical fields and diffusion and chemotaxis, which we can cover next week, depending on how far we get with the cell division. So I wanted to give a brief time to discuss project ideas. Let's just sweep the room quickly. I know several people are already working with people, but I want to just get again, you don't have to show a slide. You could just do a verbal report. Or if you want to show one slide, an update, that would be fine too. Let's just start at the top, I guess, with Tyler.
Yeah, I still want to work with the immune response to a bacterial or viral infection, and I've singled out a paper that I'm working on my preliminary proposal for. And right now I'm still thinking about ideas, like narrowing down what about the immune system I want to look at, but that's pretty much where I am right now.
Okay, so I think for that, it's important that you connect with TJ and Josh as soon as possible and have a one-on-one meeting with them to go over those ideas, okay?
Okay.
So try to set that up for this week. That's great. Drew.
Yeah, for my project, I think I settled on the paper that was on the drug delivery for like ovarian cancer, and seeing like using mathematical models to find the best way to administer drugs.
So Josh, is that something you want to talk about with does that sound like a match to you?
Yeah, I mean, I'm open to talk to discuss individual projects with people. I have my own ideas as well. So yeah, just send me an email so we can schedule what the best time for meetings are.
Oh, one thing I was going to forgot to mention is the couple of people in the class who took the class last semester. There's still a few of you who have not sent the links to your uploaded NanoHub projects, and we need those. So if you did your NanoHub project last semester and have not sent us the link, please do send it, because it's very hard for us to find it if you don't send it to us. So just as a reminder for everybody who was in the class last semester. Okay, Devin, how are your project thinking? Go ahead.
Yeah, so I'm still at the moment looking into the somatic evolution of cancer cells. And I'm looking at Professor Glazier's paper right now that he worked on some other people with. And so that one's pretty well defined in terms of the CompuCell stuff. So I'm looking into expanding that. And one thing that Professor mentioned was mutation lineage. So looking at where mutations happen and what type of cells take on that mutation and which ones don't. So it's easier to treat those types of cells. So that's where I'm going at the moment.
Great, that sounds fun. Fanbo, I guess you and Andy have been talking.
Yeah. So basically, I made several slides, and it may take several minutes.
That's okay. You're welcome to.
Sure.
Again, I don't want to put people on the spot, but I think it's important every week that we check in. On the other hand, if we take an hour each week each week to do it, it's then there's no time for the class. So we have to balance. That's great.
Sorry. Yeah.
Okay, no, this is fine. I'm not criticizing you. I think it's great that you prepared some slides.
Yeah. So basically, my project is working with Andy to develop a mechanical GUI, in the NanoHub, using the Jupyter widgets. So the basic motivation for this project is because some students may have trouble with the software distribution. So it would be easy if you can build a binary form for them to easily access the tools in NanoHub. So basically our objective is to design a user-friendly GUI in the NanoHub. So the user can do the same things as they do locally in their machine. So first I would briefly introduce several relevant methods we may deploy in our project. So the first is to introduce the architecture of Jupyter. So basically, the Jupyter contains three separate things. The first is client side, the user interact with browser to modify to run and to edit code inside it. And the server side, which the notebook, which is the Jupyter notebook server. So it's just a place where to store the Jupyter notebook files in it, and also a kernel like a Python kernel where it can run the code and execute the code and send back to the user in this kernel. So basically, I've been mentioning here is there are two different types of link here. For example, from the client side to server side, we use the HTTP and WebSockets to communicate with from the client into the server. And also from the notebook server to the kernel, we use the COMQ and also the JSON files here to connect with the kernel to run the code. And the second is why we want to use widgets to do things, because the widgets has two significant advantage. The first is it has a very good UI, for the user to interact with the like the slider here. It can be very easy to interact with the tools. And the second is the synchronization. So the widgets, the synchronization can be viewed here as an architecture. So basically the widgets architecture does not follow the traditional architecture of Jupyter Notebook. Instead, it takes the front end and the back end to connect directly using a tool called a COM. So basically the COM will skip the communication from using the WebSocket and the CRM So it will directly connect the backend and the front end like to the tools. So this is our plan. So our plan is to use the Python widget to create our custom widgets in order to fetch the image generated from the kernel, like inside the mechanical code here, and we will display the image inside the widgets. And also we want to use the mouse and keyboard events to interact with the image, so we can view and try to change the angle and try to rotate and play with the image inside the widgets here. In a future plan, so if the previous thing is done, we may switch to create a MPEG string like for a server in order to increase the speed of the widgets. And also, if we have time, we will deploy like a biological project using our new widgets as an example in the NetHub. So this is basically the timeline of what we will do in the following weeks. So I think in this week, we will, I and Andy will get set up on the NanoHub dev and trying to run the code of the Mechanica. And in the following weeks, we will first create a very simple example using the existing widgets to run our simulation. And then we will create a custom widgets, right? And then we will using the threads and interact with the user events to create to make the widgets, to make our custom widgets more powerful in this sense. And finally, we'll create a demonstration notebook. So I think that's all I have here.
Okay, thank you very much. Seeing that, which was extremely organized, I would suggest that when TJ, Josh, you follow Andy's example and Fanbo's example, and this week try to make a schedule with the students so that you actually have a week-by-week schedule of plans and deliverables, so we don't get behind. Because we're already at week 4, the semester is 14 weeks. In principle, we're supposed to have a preliminary version of these things in week 12, which means we have 8 weeks to deliver something. And so we should be trying to develop, I think, Andy, your idea of having a very orderly deliverable schedule like that was helpful. And so TJ and Josh and I and Giuliano should try to follow that to the extent that we can. I think it's a very good idea. Again, it's such a short amount of time, we have to be very ruthless about focus. Thank you, Fanbo, that was great. Let's see, who's next? Kali, I guess you're next on the list.
So I've decided, last time I spoke, I had a few ideas going around. The third one was about trying to do something with cell signaling, and I think I've decided I'm going to go that route. Dr. Glazier, you had sent me some papers, and I would be interested in trying to implement one of them in CompuCell 3D, working with Dr. Sego. And I'm thinking maybe the one about modeling dictyostelium, discodium, morphogenesis, the culmination, where it's we're looking at that stalk formation when the slime mold is generated. But I could be convinced to choose any of the others.
I think that one is a good one. We need to dig back. I have to look and make sure there are no traps or minefields hiding in that simulation. I know the one that didn't use Potts method, the one that was Othmer and who was the other person in the paper? The 3D one of flow of cells. That one they're very much related. In other words, all of those papers, basically the way that So Dictyostelium are amoeba that live by themselves. As I mentioned, they eat bacteria. When they run out of bacteria and start starving, they send out a signal and they aggregate. And then they eventually differentiate into a stalk and a fruiting body. So they have well, I mean, of course, humans have a unicellular form too, which are the sperm and the egg, and a multicellular form, which is us. The difference is that in Dicty, the unicellular form is the dominant fraction of the life cycle, and the multicellular form is a short fraction of the life cycle instead of what we have. But Dicty were very fashionable as a model for the development of multicellularity. And the thing that's quite interesting is that they go through these phases of their life cycle that look very, very different. But the behavior of the cells during each phase is not that different. And so they were a rather nice biophysical model of structure. That particular paper that you picked, the culmination one, as I mentioned, I was on Stan's PhD thesis committee. So I had some involvement in that paper a long time ago. I might start quick with the paper of Offberg because it's simpler, and the pieces you need will be there. But yes, let's TJ, why don't you take a quick look, and then let's schedule a meeting together soon to get work on that. TJ, you and Kali could meet separately, and then we could meet again after that when you've had a preliminary look. But I think that's a good project, and it's aesthetically beautiful, and it would be nice to have those working. Thank you. Great, Jeffrey.
I have some slides also here.
Okay.
So there's am I sharing the right screen, right? Yeah.
Looks good to me.
So this is the basic idea is that you can make this fluid flow from a chemical reaction occurring here in the center. And it creates these kind of convection cells because the products are less dense than the reactants are. And yeah, the papers I've read say that there are applications to biomedical devices and drug delivery stuff. So maybe in the end, depending on how it goes, I'll be able to tie it back to biology in some way. But right now we're focusing on the fluid flow. This is just some basic concepts that will, for people not familiar with fluids, I guess. I guess the most important one is the no-slip condition, that particles in contact with the boundary don't move relative to the boundary. So first, we're going to look into simulating lid-driven cavity flows because it's relatively common first step in modeling fluid simulations. So that's what we're going to look at here the next few weeks or so. And we're also going to look at Poisson flow or Poisson flow, which is a kind of standard test for testing flow. It's a little more complicated though, but if we can get that to work, it'll be a step in the right direction. And convection cells are also what we're looking at. This is the same picture from the paper again, where this is the chemical reaction they have. And it's exothermic, so there is a little bit of heat. They don't go into much detail on the heat influence. They focus on the buoyancy effects in diffusion more so. So I'm not entirely sure how much heat plays a role, but yeah, since convection cells are the basic flow patterns here, if we can get that, then we'll be able to add the chemical reactions in, and yeah, then we'll basically have the main components we need to simulate this. And yeah, that is all.
Great. Well, that would be certainly would be very nice if you can, if Foundbo has the user interface working for Tellurium, and we have that, that'll be pretty exciting. That'll be a great demonstration. Now, NanoHub wasn't originally designed for doing biology. It was designed for doing chemical engineering, essentially. And so your project would fit perfectly with that kind of approach. So it'd probably be very popular if you could get that one done. That's very nice. Okay, I guess it's Furkan. Is you the last of the list? Have we gone through every? If I missed somebody, please let me know.
Yeah, also I have prepared some slides to share.
Okay, sure.
So actually this semester I decided to work on reproducing model with two different approach. The first approach is the Composite 3D, and the second one is more center-based one, which will be the Mechanica. In the last last class, actually I prepared a kind of a composite 3D model for the intestinal crypt. And in this crypt, actually, what you will see is there were two different type of cells, and those are actually the fate of the cells are kind of controlled based on two signaling network, which is the WNT and Notch. And there were some stem cells at the bottom of the intestinal crypt, and then those are making a proliferation, and they were pushing the other cells to the boundaries, and then according to the there is another signaling pathway what we call delta notch. This is a kind of a contact-based signaling network. So roughly when one type of cell is having a delta high and the other one is delta low and notch high, just roughly explaining. And so the how it looks like this, actually I don't have the animation, but what you can see is that the delta levels of the cells are high in here, and the notches are vice versa type, and the type of cells are kind of like found type of cells are calculated or predicted according to those two different one, and the WNT is a kind of a gradient, high at the bottom and low at the top. And so for this one, this is kind of ready. What is missing, what is missing, the WNT is not secreted, it was fixed, so it will be added one feature that the bottom cells will secrete some delta notch and I'm sorry, the WNT, and to see the first thing that there will be validation of that the gradient is still true. And for the Mechanica implementation, so last week actually I showed this one, but the approach will be kind of different in here. So we will use a cluster approach. What you will see in here, you can see there are two type of two cells, and those are actually again particles, but particles make a cluster, and those clusters will represent the cell, and they are dividing, as you can see, and those clusters will be used in the model. So yeah, and what so yeah and also the second thing will be the important thing, the flux feature in Mechanica, and in this flux feature, so what we can see species are maybe that might be WNT value or delta not the delta notch. The WNT will be flux implemented, so there will be flux gradient over the domain, and so for the because we are using cluster approach, which will be representing our cells, we will use the mean values for the clusters, and delta notch signal also will be implemented in here, and those calculations will be on LibRoadrunner, I guess, and we'll use Mechanica will use LibRoadrunner to calculate the temporal model in their subcellular level, and yeah, and the LibRoadrunner will use actually SPML, which is the systems biology markup language. And the timeline is for this week. The morphology will be important. The intestinal boundaries, the cylinder at the like middle, and the next 2 week I will work on with Randy to make some morphological trials to confirm that lozenge object is true, and like the movement of the cells and the division and cleavage of the cells and split of the cells, and also the cluster approach will be examined during these 2 weeks, and the next 3rd and 4th week the signaling network will be implemented, and if the LibRoadrunner will be used in the Mechanica, and to make the WNT, not the WNT. WNT will be just flux, but the delta notch will be SPML related. Yeah, thank you.
Great. Okay, I think one thing to think about, I mean, again, the model you had was a very nice one, but it might be worth checking to see if there were any new ideas about how this signaling is happening. Because I know Arthur Lander, whose brother has just become the director of science for the government. But Arthur and Natalia Komarova had done some work on trying to understand the signal regulation. I'm not so sure. I thought they did colonic crypts. I know there were people at Irvine who had some ideas about signaling and regulation of the colonic crypts that were slightly different from the model you were working from. There's this wonderful video of Arthur giving a talk in Banff in 2019 that I've recommended. He's talking about regulation in the context of cancer, but there's a big question, which is when cells divide, how do they know when to stop? In other words, when you grow, there's no signal set from your left arm to your right arm in your embryo, and yet your arm lengths are within a few percent of each other. And that means that size regulation in biology has to be pretty accurate. It's not perfect. Different snakes of the same species, even genetically identical individuals, clones, have different numbers of ribs. So these kinds of things are typically accurate to about 5%. But given the noise in the systems that are generating them, the fact that they're good to 5% is remarkable. And so Arthur has been looking at the mathematics of how you get very accurate size regulation. And the base model, which we have here of the colonic crypt, which is you have cells at the bottom that are stem cells, they go to some preliminary state, then you have transient amplified zone, and then they differentiate. There's some problems with that in terms of, not that it doesn't work if you fine tune it, but that particular model has some problems if you have noise in terms of its regulation. And actually, we know colonic crypts have some problems in terms of noise, because that's why you get polyps and eventually get colonic cancers. So it's not a perfect system, but the regulation works a little better than you might expect given the way that model works. And so I would just check at what Arthur and Natalia have written recently about this, and see whether, no problem replicating the model you had from the last time, that's fine. But it might be interesting to see, and relatively easy to put in some additional hypotheses about the regulation structure. It's only a few lines of code, really, and you could then compare that. The other thing is that if you're doing it in Mechanica, you could have more of that at transient amplification. In the CompuCell, you had relatively limited cell division because pushing things takes a lot of time. But in Mechanica, where you have solid mechanics, you could have rapid cell division. And so you might be able to build a slightly more realistic in terms of number of cells. So I think it's a great project, and having those two back to back, I think, would be very informative. And of course, you'll all benefit if Fadbo gets the user interface. Although you wouldn't need it. In other words, you wouldn't have to have the user wouldn't have to be able to do mouse clicks to steer. You could still do it in other ways. I know, for example, Jeffrey in his little app from last semester had a 3D plot using Matplotlib, and then you had sliders that let you rotate it by pulling on the slider. It's not as elegant as hovering over it with a mouse and being able to manipulate it that way, but it worked pretty well. So you may have some backups on that. These are all great projects. And again, I think we need to try to come up with these kinds of strict schedules. The schedules could slip, but at least an aspirational schedule so that we make sure we deliver. Great. Did I miss somebody? I think, is that everybody? If I missed somebody, they please speak up. Okay, great. Thank you. So the people who've already met with people to help, good. If not, make sure we meet. Everybody has at least a meeting before the end of the week. And I think trying to meet twice a week, at least once a week, and ideally twice a week, one-on-one with people is good, because otherwise it's going to be hard to make things work in time. If it does work in time, that's fantastic, because we have some good projects here. Every one of these is a great idea. Okay, thank you, everyone. So, and now we'll come back to the lecture, if that's okay. Is there anything, well, maybe I should say, is there anything else people want to discuss before we continue the class? Are people okay with this model of having a brief check-in, group check-in at the beginning of class? Does that work for people? I don't want it to be too long, but I think it's useful for people to hear what other people are working on. So we'll see how we can adjust things as time goes on. But I think it's good to have people have a sense of community. Okay, so last time, the first thing we did was we talked about this in CompuCell, the idea of a steppable. And this was a Python class that does something. And it's quite possible to write all of your Python CompuCell code into single steppable. Maybe later in the class, we'll talk about multiple steppables and some of the issues with that. But the key thing about these steppables is that they have a particular computational architecture. There's some boilerplate at the top, which you can ignore for the moment. The name of a class, which the CompuCell uses to register the steppable. There's an init, which you always get. And again, most of the time you can ignore that init, except perhaps for the frequency equals 1, which means it's executed every time step. If you made it 10, it would be not 10 times per time step, but once every 10 time steps. So it should be period or interval rather than frequency. There's a start function, which is called once at the beginning of the simulation. which is usually used to initialize things. A step function, which is called periodically. And it does the main part of the work usually. And then a finish function. And often we interrupt these programs so then the finish function isn't called. So we don't tend to use the finish function very much, but it's not a bad idea to have it there. And if you're running batch mode on clusters, it's often useful to have it. One thing you'll notice about Python, of course, is indentation. And you'll see the indentation here because you have a hierarchy of functions. Okay. The main thing that we're going to be working with inside of CompuCell are cells. Cells are Python objects, and they have a whole series of attributes, some of which are automatic, like cell.id or cell.type, others of which occur because you've loaded particular plugins. Cell.target volume, for example, cell.lambda volume. And the editor, Twittit, has boilerplate code listing all of these things. So if you don't remember the syntax, you can get it there. As I said before, Twittit, usually with Twittit, the biggest problem is finding where things are living, because there are a lot of options. And sometimes the most common ones aren't in the, it's a little bit like Microsoft, the most common ones aren't in the most convenient places in the menu hierarchy. Something that we will do a lot in CompuCell, or for that matter, Mechanica, is do things to all cells or all objects of a particular type. Inside of CompuCell, the thing that you're going to use the most often is something called self.cell_list, which is an inventory of all the cell objects in the simulation. And so we will very often be doing for cell in self.cell_list. You'll get very tired of seeing that colon. Remember that in principle in Python, when you iterate over a list, the order isn't guaranteed. And you also don't want to do things that create or destroy elements of the list while you're iterating over it. And that's going to come up a little later when we do division. You don't want to be iterating over a list and modifying it at the same time. And there are a couple of iterators. Some of these iterators are things that we're not going to use very often or at all in this course. For example, CompuCell allows you to build, Furkan was talking about these cells that were composed of lots of particles that were aggregated into a cell. CompuCell also has a concept like that, where you can divide a cell into subcompartments for more spatial detail. It's possible that some of your projects will use that, but there are special iterators for iterating over the components of a cell. The ones that you'll use pretty frequently are iterate over all cells or iterate over all cells of a given type. Sometimes you want to iterate over all pixels in the lattice. In general, you don't want to do that in Python. It's slow to do that. Iterating over cell boundaries can be very useful. Iterating over cell neighbors is something that we'll do today. That's something we need to do. And we'll find that the object structure for doing that is slightly confusing. At least I find it slightly confusing. So we'll come back. Okay. Something that I meant to mention last week that I forgot is that when you create cells, every cell has a unique ID, cell number 1, cell number 2, cell number 3. It also has always a type. And we created in the XML a type, a list of cell types, medium, mesenchyme, cell number 1, and so on. In the old days, you had to refer to cell types by number, so you had to remember them. Instead, it's more convenient to identify cells by a named type. And so you use a self.type name. So if the name of the cell type was mesenchyme in the XML, it would be self.mesenchyme in Python, with the caveat that you have to put it in all capital letters. I'm not exactly sure why. If TJ's on the call, maybe he can explain why why we have a convention that it has to be all capital letters, but it does. That's the one thing that you have to. But if here's the iterator over cell list by type. So for cell in self.cell list by type, and then you list the types. And so you could iterate, suppose you had 10 different cell types, you would iterate over 5 of them or one of them, you put them in the list. But anyway, self.type name with the capital letters gives you that thing. And we also then introduced the idea of scientific plots. I hope people in their homework were able to play with those a bit. Fundamentally, there are 3 lines of code you need to know to be able to create plots, arbitrary plots in CompuCell. You have to create a window that lives in player, self.plotWin, self.add new plot window, opens a window. Self.plotWin is a handle. It could be self.artichoke. The name doesn't matter. It needs to have a title. The title needs to be unique. I'm not exactly sure why, but it does. You can't have two plot windows with the same title. X-axis needs to be named, Y-axis needs to be named. You can change the scales to be linear or logarithmic. You can have grids, and there are a variety of other options. And so this is basically the panel that you see. It doesn't have any data sets in it. The second thing you have to do is add a data series. And so you have the name of the data series and the style. And again, there are a bunch of options for lines, dots, and there are others, colors, size, and so on. These usually go in the start function because you create the windows at the beginning of the simulation, but they can be used elsewhere. If you don't want the windows to appear until 100 time steps into the simulation, you could put them into the step function and have them execute at time 100. And then inside of a step function, you actually do the addition of the data set. Sorry, the points to the data set. And it's the handle self.plotWin from here. Add data point, the name of the data series to which you're adding from here, and then X comma Y. Here, typically X is the time, but it doesn't have to be. You could be plotting 2 variables against each other. You could be plotting X position versus Y position and have a trajectory. So you have a lot of options. And there's some options for blanking the screen, for redrawing things, which don't get used so often. There also are equivalent start and step functions for plotting histograms. And if you're interested, you can look at the histograms in Twit and in the manuals. But they're pretty easy to set up. So the display is pretty good. You can save the data from these plots. Actually, if you right-click on the plot, you can save the data as a CSV or an image during the run. And you can also write code that will do the same thing. So the first homework problem was to generate a 3D simulation with 100 by 100 lattice with a single cell with surface and volume constraint. Sorry, I said single cell. So 1 cell type, we called it cell 1, could call it anything. And then we want to draw a blob initializer, create a sphere of cells at the center of simulation, and then set the target volume and surface to what they are here. To begin with, it's a target volume and target surface 225, lambdas of 2, and create 2 plot windows, one for cell volume and one for cell surfaces. And then plot the individual volumes and surfaces. So this could be quite a few data points. And then after you got that working, the question was to add a little code to plot the mean and volume and surface versus time. And how did that go for people? Were there problems, or was that, I can just sort of, before I do anything else, I'd like to poll people. I could spend a few minutes going over this, depending on how many people need it. So maybe I should just launch a poll. There's my poll. Where did my poll go? Poll. So in the poll, if you did it, if you were able to do this exercise, do it at home, just click finished exercise. And if you'd like me to go over it in a little more detail, click need help. Okay, and get a sense of what people would like. Because I don't want to take a lot of time on it. If you could, everyone could do it. On the other hand, if you want me to go over it, okay. Okay, looks like everybody was okay with that. So in that case, I'm not going to repeat it now. That's great. I'm glad that worked for people. The next thing that we didn't really talk about in class, but was mentioned and was in the homework, was that to begin with, we defined the volume and surface constraint in the XML. And that's wonderful if you want all of the cells of a given type to have the same volume and surface. And in many cases, that's what you want. But if you want cells to be able to grow, then each cell has to have its own target volume and its own target surface area. Otherwise, you can't control them individually. We could do it together. We could go into the steering panel and change the target volume for every cell of a given type, and all the cells would grow. Or shrink it, and all the cells would shrink. And the homework explored a lot of that. But in reality, cells grow and shrink independently of each other. They don't all do it together. And so that problem 3.4 was a little bit of a stretch exercise to work out some things that we were then going to focus on a little more in this class. And this is about how you can manipulate volume and surface area for each cell independently. They used to have the volume and surface plugins used to have different names for the local one and for the global one. Nowadays, they just have the same name. And the syntax is still basically the same. You have the plugin named volume. And instead of specifying the parameters the way we did in the XML, you just have the volume plugin. You say plugin name equals volume, either end plugin or put the backslash inside of the tag pair. And that, when you do that, it tells CompuCell that it needs to load the energy functional. So that still has to be there in the XML. But the fact that the variables are not specified in the XML tells CompuCell that those are going to be specified in Python. And so if you create plugin name volume, plugin name surface, and that's all you do, and you try to run it, the cells will disappear because those parameters weren't specified. So what we're going to have to do then is go to our Python in our start function. And in the start function, we'll say for cell and self.cellList. In this case, we're going to assume that all of the cells, whatever the type they are going to have the same target volume and surface. And we iterate over all the cells in the list, and we say cell.target volume equals 25, cell.lambda volume equals 2, cell.target surface is 50 here, cell.lambda surface is 2. So that would be equivalent to having said inside of the volume constraint, target volume is 25, lambda volume is 2 for all the cell types. Target surface is 50, lambda surface is 2 for all the cell types. Clearly, we could be more flexible here because we can control it at the cell by cell level. The one thing that we're going to face which is a little less convenient when you use the Python specification. In general, the Python specification is easy and flexible. The one thing you'll run into a little bit is that we've done steering. In other words, we use that window on the left-hand side of player, and we could open that and change parameter values like the target volume or the target surface area that we had specified in the XML and see the effect as we were running the simulation. Parameters that are specified in Python rather than XML are not available in that left-hand menu. That left-hand menu really is a parse of the XML. And so you don't have the ability automatically to change parameters that were specified in Python rather than XML. And now, a year or two ago, one of the big improvements to CompuCell was that there was a widget added that allow you to open a steering panel like the ones we saw earlier for Jupyter. And so you can then have any parameter in the Python steered through a graphical user interface. But you have to do that by hand. In other words, you have to create a steering window, tell it what parameters you want to steer, and then have that available. But you can do that, and we'll cover how to do that a little later in the class. Not today, but in a later lecture. Another thing that's important and very, very useful in CompuCell, again, it wasn't something that was added until relatively recently in terms of the history of CompuCell, is that we discussed that the cells have attributes like ID, volume, surface, target volume, and so on. Every cell comes prepackaged with a dictionary. And what's the use of having that? Well, the dictionary is basically a sort of, you think of some kind of role-playing game. It's like a sack that you can carry. You can stuff anything in it, and like a sack in a role-playing game, it doesn't get full. You can stuff as much stuff in it as you want. So if I want to create an attribute for a cell to hold some information that isn't already available, I can create a dictionary entry. Cell.dict references the dictionary for that specific cell. I can create any variable name I want, quote, attribute name, and then set it equal to some value. And the great thing about Python and Python dictionaries is that they're completely flexible in terms of what I put in them. So one dictionary entry could be, unlike arrays or any kind of standard numerical object, the Python dictionary entries are completely flexible. One, I could create cell.dict quote name equals hello, where it's a string. Cell.dict quote value equals 10.6, a floating point number. Cell.dict list history equals square bracket 10, 11, 13, 15 is a list. I could even put a dictionary inside of the dictionary. And so I can attach any information I want to a cell, and we'll find later that that information also is carried over when cells divide automatically. And so this is a very powerful function. You can extend the object class, the object cell, using the dictionary to do any storage that you need. Something we didn't have until recently, but that we'll come to later, is that there also is a dictionary that lives inside of CompuCell as a whole that you can use to pass information, and we'll explain why you need to do that a little later. Has everybody in the class worked with Python dictionaries before? Is there anybody who hasn't run into a Python dictionary? So this is, I mean, syntactically, cell.dict quote variable name close quote equals is a little bit longer. It's a little more verbose than cell dot name, like name, but conceptually it's no different. If you attach something like a list, if you use make a dictionary entry that's itself a list or a dictionary, then you have to use the standard Python you can use the standard Python slicing operators, append operators, remove operators, average operators, all those things are available. Okay. And that really was coming about because in that homework problem 3.4, homework problem 3.4 started out by saying the pressure in a cell was defined as -2 times the target volume, times lambda volume, times the target volume minus the actual volume. That is that the cell is behaving like a balloon, an ideal gas balloon, where if the target volume equals the actual volume, there's no pressure or tension on the surface. And the membrane tension is defined similarly. And so this request was that you create for each cell an element of the dictionary, which we call pressure and surface tension, calculate those pressures and surface tensions, and then plot them. And then you could put, for example, create a loop. In the homework, the rate of growth that we asked you to do was crazy, and we'll do some rather strange things. Increase the target volume by 5 every 10 MCS. That's actually a lot, and it'll misbehave. 0.05 might be better, or 0.5 might be better. And then see what happened. Were people able to do that homework? Oh, again, I can relaunch the poll, and we can work on that if we want. Okay, so on this one, I think people will need some help. So let's try to do this. So let's all try to do this together. I will try to just do it. I'll do it raw. Let's do new CC3D project, and we'll call this, I don't know, pressure check. I haven't. Josh prepared a code to do this, but I'm going to wing it. Let's try this. NanoHub. Untitled folder should be okay. And we said to do it on a cubic lattice. Let's make it 128 cubed. Did I say to do periodic boundary conditions? I don't remember. Make the pixel copy range 2. We'll use blob initializer. And we're going to have just one cell type. I said to call cell 1. No chemical fields. And we're going to have volume. And this time we're going to use the local flex, which is just gives us the volume and surface without defining them in the XML. And what else do I need? Do I need center of mass? No. Cell neighbors. We'll need that in a minute, so let's turn that one on. PF. I think that's it. Josh, if I make a mistake, let me know. Okay. So we'll do that. Why doesn't everybody try doing the same thing? I can go over that again, but does everybody have a Twit running, and can you do that? So the first thing to do is create a 3D lattice. 128 by 128. Put a cell in it, cell type in it, and then create that. And I'd like everybody to do that together. And the one thing we're going to have to do in our XML, well, actually, radius of 25 is fine. Everything here looks okay to me. And let's see what happens if we run this. Not very pretty, but all right. That's what it does. Probably because the volume, the target surface area is too big for the volume. So I'd like everybody to try that. So I'll give you time. Can everybody try? Can everybody do that? It's useful to try to do that. Oh, I did what I said I should. I did what I forget to do. Having said that, you have to specify the target volume and the target surface area in the code. I didn't do it, so we'll figure out how to do that in a minute. No wonder things are a mess. Not very pretty. But that's the XML. So I'll give people a minute to catch up on that. Sorry. Better mute that. You don't need to hear me drink. Okay, I'll give people another minute or two on that, and then we can move ahead. Notice here, volume, plugin name, volume, and surface. There's just the tag name is empty. If anybody needs help, raise a hand. Okay. So now we're going to have to specify the target surface and volume in the Python. We go to the Steppable. And which component of the Steppable do we do the specification in? The initial one. Somebody? Where does it go? Well, if we're going to specify the initial target volume and target surface area, we put it in the start function. So I could put my cursor here in the start function. I go up to CC3D Python. I do visit all cells. Okay. I don't actually want to print the information there. And then I want to set the target volume, surface area, and so on. I can go to CC3D project. I go to cell attributes. And I want to set the lambda surface. I want to set target surface. I want to set target volume, lambda volume, and I want to set target volume. And this is actually reading it. I want it to go the other way. So I want to set that. So I'll set cell.lambda surface. We were using a number of 2, I think, 2.0. Cell.target surface is the area. And I think we had said something like 25 before. No, bigger, 50. Cell.lambda volume is 2. Cell.target volume, we had 7 by 7 was about 125. 125. Something like that. Try that. Save it. And now go back, stop. See what happens. If the screen doesn't read raw now, the cells aren't breaking up. Now you're getting compact cells. So why doesn't everybody try that? See if that works. I'll show you. I'll go back to the code. So this is initializing the target volume and surface area. And while we're at it, let's create, well, I'll wait a second till people catch it. We'll create a dictionary entry for the pressure and the surface tension to keep track of those. And we can do that here in the same loop that we just used. Again, if we don't remember the syntax, cell attributes, access modify dictionary attribute reminds us what that is. I'm going to delete some of the comments just because there are too many of them. But we could create a cell dot dictionary attribute named, say, pressure. And the pressure was defined to be minus 2.0 times cell dot lambda volume times cell dot target volume. Furkan, what's up? Go ahead.
So in the example, or when we get the dictionary, sample dictionary, the attribute name is capitalized. Should it be capitalized, or it doesn't matter?
No, that was doesn't have, no, it doesn't have to be.
Okay, thank you.
It's Python though, so that if you capitalize it, you have to be consistent. I think that the type name is the only one that's strange that way. In terms of having to capitalize something that's not capitalized in the original. But this is just a regular dictionary entry, so you're free to use any name. And we want the pressure as well. I mean, sorry. We want the surface tension as well. So we'll do this. Okay. So now I've got the pressure and surface tension for every cell. All right. And that's something now I'm going to have to update that because that will change. So let me copy this, and in my step function, actually I already have the for cell and cell dot list. I'm going to have to update those values periodically. So if I want to update them, I have to put them in here. I have to make sure my indentation is right. And so this will recalculate. This will recalculate the pressure and surface tension every time. Okay. So that gives me the pressure and surface tension. And now let me put in the plots. I don't want the plot to be inside of a loop. I want the plot to be outside the loop. So I have to egg out that. CC3D Python, scientific plot, setup, self.plot window, and we can do pressure and surface tension because I want to display those. X-axis is still time. It's okay. Y-axis is linear. Fine. And let me just do this one is pressure, surface tension. And we'll use dots because there are a lot of them. And we'll make the size 2 maybe. So this is now creating a place to plot those pressures and values. And the one thing I may find out is I may have got the sign backwards, so I may have to change this. And then inside of my loop here, once I've gotten my pressure, I need to add my data points. So I go back to CC3D Python, scientific plots, add data points. And I go do the values from here. So pressure and surface tension. And instead of cell volume, I have to grab cell dict of pressure. And cell dict of surface tension. So why don't people try that? Is it clear what I've done? Let's see. You want me to try to see if it works before you try repeating it, or do you want? No guarantees that I haven't made a typo. Let's see what happens. Stop. Run. Oops. Typo. What was my typo? It says let's see, what did I do wrong? Invalid syntax. That looks like probably a parenthesis off. Huh? Let's see. Does anybody see what I did wrong?
I guess lane 57. That surface was not written
59.
correctly, I guess. Lane 57.
Typo.
Yeah. 57.
Okay. $57. Oh yes, try capital.
And also at the end, it's like the cell.sorfa. It lasted like this.
Oh, okay.
At the end.
I cut and pasted, and I lost the tail. So I hope that gives people time to catch up while I'm correcting my mistake. Let's try that. Okay. Average pressure surface tension already exists. Please use a different name.
I guess I got that error too. I guess in the start function, the indentation of the plot should be one left.
Oh, these are, yeah, sorry. I said to make sure that they're not inside the loop, and then I didn't check. Okay, good. And it's annoying you have to joggle the screen each time. There we go. And now remember, use tile to lay things out. Okay. So it's not the most informative thing in the world, but there it is. I'm not sure which color is which. But you notice if I right click, I can change the scales and do variety, export the data set. All right. Okay. So now I've got my pressures and volumes plotted. And now I could do, I'm not going to do all of it, but now I could do one of these exercises. So increase the target volume by 5 every 10 MCS. I'm not going to do that. I'm not going to do it that fast. But inside of this loop, I could say, if doing things periodically in Python has somewhat, there are many ways you could do it, but Python has this funny syntax, which is integer division. So if I do if not MCS %10 colon, that'll do something every 10 Monte Carlo steps. And so let's just do what we asked for, which is increase the target volume by, not by 5. Well, let's do it by 5. So in that case, I do cell dot volume, target volume, plus equals 5. And let's see what happens if we do that. Nothing good, I should say. All right. I don't know why it won't let me joggle. Huh. Let me try that again. There. Okay. Now I can see my cells. And let's see if the target volume actually goes up. Yeah, you could see the bottom here. I'm changing the target, and you see every 10 Monte Carlo steps, there's a step here. And that step is happening because the target volume is changing, but the actual volume is not changing with it. And so it's pulling the pressure. Okay. So I don't want to do the whole exercise, but does that give people I'll go back to the code for a second. Does that give you the basics for what you need to do this exercise? I can repoll and see if that makes sense. If people want me to go in more detail, I can. But I wanted to show people the basics of how to do that. There's one other thing that I guess might be interesting to do, which we really should create a new plot window for. but I'm going to be just quick and dirty about it, which is suppose that I get rid of that second plot, and I'm just going to call it pressure for the moment. And suppose that instead of plotting pressure versus time and temperature and surface tension versus time, it might be interesting to plot the pressure versus the surface tension. Maybe they're correlated. I want to see how correlated. So the people who are working on this can just keep going, but let me try that. Well, clearly something is wrong because one of these values isn't being updated, right? It's always 976. I wonder which one is wrong. It's not doing what I said. So one of these two is not being updated properly. Lambda volume times cell target volume minus cell volume. Lambda surface times cell target surface minus cell surface. Do I have a typo somewhere? Oh, I know, because I didn't have, it wasn't consistent with my capitalization. I used a capital T in one place and a lowercase t in another. Makes you nostalgic for Fortran. Maybe not. Let's see if that works. There we go. And as promised, there's an interesting relationship between the volume and the surface tension. It's not just a random scatter, and it's not just a single point. So the ability to plot variable against each other is quite useful. Any questions about that exercise or how to do it now? Again, I'll show my code again. But one of the things that's most convenient about CompuCell is its ability to do plots. So it's not that you can't make mistakes as I did, but it's also not that bad to do. And that ability to generate plots on the fly, I think, is really one of its strengths. Josh, did you have anything else on this you wanted to cover?
No. I think this is fine. I just in terms of manipulating the plot properties, just remind people that when the plot is created here in the add plot window, and we'll cover a little bit of it later on, there is multiple, and in the add plot, there's multiple options that people can manipulate in terms of coloring, the style, whether or not to turn on or off the grids, and also whether or not to turn on the legends. That there's gonna be a little bit of a snippet later on during the course, but that also helps in terms of manipulating the data and just portraying the information in a useful way.
Now, some of these, the right clicks on the, when you're hovering over the plot, it gives you a bunch of options. The auto scale and things, those work. The plot options here, most of those don't work. So if I select transform, it'll offer me a Fourier transform that doesn't work. So I believe the grid should turn on. So I should be able to turn on the grid. Yeah, the grid I can turn on with the mouse clicks. And so most of these, some of these work, but I could also save the data set if I wanted to as well, either as an image or as a CSV. But I'll warn you in advance that some of the fancy features there don't work. The package that we use to do the plots has some built-ins which are a little dodgy. They haven't been turned on. Okay, there's still a few people who say they need time, so I can wait. Give you another. If you finish the beginning exercise, you could try doing B, C, D, and see what happens. But I think it's important. Again, once you've done this a few times, you'll find you're doing the same thing again and again and again. But getting the hang of it the first time is not trivial. And so I think it's important to take a little extra time now to make sure that everybody can build some of these basic functions. And once you've done it a few times, it gets to be. Doesn't mean that you're not. As you see, I can type something wrong. We are always able to do that. But thinking about the structure of the program, I think the basic functions that you use pretty frequently. So we've shown how to create a dictionary entry. We've shown how to do a plot. We've shown that you can plot against time or against a value. How to calculate pressure and surface tension. So it's 6:30. Why don't we take a 5-minute break, and the people who are working on it can keep working on it. And the people who are done can take a break and come back. And as I say, if you have done the basic exercise, you can try playing with some of these other examples by changing the target volumes or the target surface independently and see what happens. There's actually a lot of depth to this exercise. If one explores it in detail, it could take quite a while. If anybody needs more help, please come back and feel free to speak up. Okay, so the other thing that was in the homework, which is a little bit less convenient in the 3D, so I don't think we're going to go into it in detail now, but it's again a convenience feature that's very powerful in CompuCell, is what's called a tracking field. And this one is a little bit surprising because it has to go in the init function, not in the start function. And what a tracking field does is very often if the cell, it's a little bit like doing fluorescent vital labeling, fluorescence labeling in microscopy. So very often you might have a scalar value like the pressure of a cell. And we want to be able to visualize that. And we visualized it here by plotting it on an axis. But what we might want to be able to do is actually see the cells colored by the pressure. And in the old days, we had a whole variety of things that we had to do that. But in fact, CompuCell offers you the ability with one line to create an image that is the cell field colored by the value of any dictionary entry. And so let's actually, well, maybe we should do it. So let's try this. So here we have a tracking field. We have self.track cell level scalar attribute, field name, and then attribute name. And attribute name is the name of the dictionary entry. So if I go here into the init, and I go to CC3D Python, and I go to, let's see, extra fields, automatic tracking. And I could actually track a vector and it'll plot a vector. But anyway, let's do track cell scalar attribute. And I'll give the field name. A field name, we'll call it, say, pressure. And the attribute name has to be exactly what the dictionary entry is. The dictionary entry was pressure, lowercase pressure. Pressure. And let me make sure that I actually put it in lowercase everywhere in my step function. Pressure. Updated here, pressure. Good joggle. Sorry about having to continually re-joggle the screen. Yeah. So let's try that. I'm going to run that. And at first you'll say nothing, there's nothing there. Where is this extra field? Let me hit pause. If I go to cell field, you'll notice there's now an extra entry called pressure. And if I hit this button here, this two window button, that will create an extra window. Like that. And I created two of them. I don't need two. And this is cell field. I tell it to display pressure. And each cell will be colored by the numerical value of the pressure that's attached to it. Is that reasonably clear? That's actually a pretty powerful thing to be able to do. I don't actually know what happens if you try to render that in 3D. I don't know if it works. I don't think it does. No. If I try to do it in 3D, it doesn't work. It only does it. And so if I have something like the phase of the cell cycle that I want to represent any numerical value that's associated with each cell, I can use this tracking field in one line to render it on the screen. So now I'd like to do a classic simulation, which was where this whole project started 20-some years ago, which is cell sorting. So in many tissues, and a classic example of this would be in the eye, but it also shows up elsewhere, you see layers of cells. You see a layer of cells of one type on top of a layer of cells of another type, on top of cells, a layer of cells of a third type, and so on. And in early embryonic development, if you take those cells that normally form the layers, take, say, 2 layers, and you dissociate them into single cells, and you mix them up randomly and make a blob out of them, and you keep those cells alive, what you find is that one of the 2 cell types will cluster together, and if you wait long enough and the cells survive long enough, they'll sort out. So you'll get all of the cells. In this case, these are from the eye. The black cells are the pigmented cells at the back of the eye. The transparent cells are the cells that are going to form the photoreceptors later on. All the black pigmented cells come together to form a clump, and all of the progenitors of the photoreceptors clump together and surround the other cells. And you could do this kind of experiment with multiple cell types. For example, you could take cells from the limb of the chicken, this is a chicken, and mix them with these pigmented black cells from the eye. Of course, that wouldn't happen in reality. You could mix the eye cells with the heart, and the heart cells with the liver, and the liver cells with those unpigmented neural retinal cells. And you find that there's a consistent hierarchy, that the red cells go outside the green cells, the yellow cells go outside the red cells, the blue cells go outside the yellow cells, and so on. And that suggests that there is some property of these cells that's consistent from cell to cell by type that determines how they move. And if you look at this rearrangement, it looks very much like fluid droplets. It looks like if you mix oil and water together, the oil will separate out and coalesce into droplets. And the differential adhesion hypothesis, which was developed by Malcolm Steinberg back in the 1950s, late Malcolm Steinberg, he died a few years ago. He was a professor at Princeton, one of the developmental biologists at Princeton University, wonderful man, said that maybe what's happening is that the cells really are behaving like a fluid. And the thing that's determining this sorting out of the cells is something like surface tension. And his hypothesis was that cells have a consistent adhesion to each other. And the fact that they stick to each other consistently with a little bit of random movement leads to this kind of separation that we see. And the cells that go to the middle of the clump are the ones that stick more tightly to each other, and the ones that go to the outside are ones that stick less tightly to each other. And this was called the differential adhesion hypothesis. Now, in reality, there are other things that could be happening. Differential adhesion is one possibility, but the cells could be pulling on each other actively through their cytoskeletons. And so that could be differential contractility. The cells could be sending out a chemical signal and moving up or down the chemical signal. All of these could give rise to the same sorting pattern, but the kinetics the time course of it would be a bit different. And over the years, it's been shown that while differential adhesion is part of what's going on, it's not the only thing. That depending on the particular situation you're in, all of these other mechanisms can be there in addition to adhesion or instead of adhesion. But in this particular classic example, where you take the cells from the retina, the neural retinal cells and the pigmented retinal cells, it really does seem to be differential adhesion that's going on. And this is where we started back in 1992. I guess I met Mel then. These are actually experiments. I don't have a wet lab that's operating anymore, but I used to do quite a few experiments. This is actually experiments from my lab a long time ago in chicken. And these were some of the first CompuCell simulations that we did, long before there was CompuCell. These were 3D simulations and a precursor to CompuCell, run on an ancient computer called the Sun 2. And one of the questions we asked was, what kind of clump sizes were there as a function of time? And so this is the simulation that I'd like to do together with you today. And the basic thing that we need to add to what we've done so far is that we've given cells volumes and surfaces, but we haven't really said how they stick to each other. In reality, depending on the type of cell that is in question, cells adhere through all sorts of relatively complicated means. And in particular, the adhesion between two cells is not constant in time. If I take two cells and bring them into contact with each other, they'll stick, but the degree to which they stick will change in time. And that's not going to be included in this model. The cells have a whole variety of cell adhesion molecules that cause them to stick to each other. Depending on the cell type, those adhesion molecules can either be distributed more or less uniformly over the surface of the cell, or they could be localized in bands, or they could be localized at points. And there are certain molecules, molecular species, like cadherins or CAMs, that primarily connect cells to other cells. And there are certain molecular species like integrins that primarily connect cells to non-cellular material like extracellular matrix. When cells adhere to each other, those adhesion molecules, besides sticking to each other, also on the intracellular side of the cell send signals. And so that is, they're not just passive physical objects. They do much more than that. And they also bind on the cytoplasmic side to the cytoskeleton and cause the cell to rearrange and do things in particular ways. They also in themselves rearrange in the membrane. And when they do that, they tend to form stronger junctions. And so the adhesivity between cells tends to go up as they stay together. You could almost imagine if I put glue between two surfaces, the surface tension of the glue will hold them together. But when the glue sets, I get a very strong bond. And that is a little bit like what happens. The cell adhesion molecules start coming together. Once they've bonded, if they stay bonded for a long time, they rearrange to form a much tighter, more permanent junction. Those junctions are never completely permanent. You could always do things to disassemble them and reassemble them. Another thing that's a little bit counterintuitive, which we're not going to model here, is that if I have two cell adhesion molecules that are joined together, and I pull on them, what's the probability of them detaching? And normally, for example, if I have a surface with a piece of adhesive tape on it and I pull on the tape, the harder I pull on the tape, the faster the tape unrolls. So the breaking of those bonds in the tape, the rate of breakage goes up as I pull harder. Rather counterintuitively, a lot of biological molecules, the bond breaks more easily when it's not being pulled on than when it is. And the biophysics of how that works is rather complicated, but it has a sort of a paradoxical effect. For example, in the bloodstream, if I have a blood cell that binds, that sits on the wall of the blood vessel, and there's no flow, I could very easily displace that cell. If there's flow, so that cell is being pulled hard, the strength of the bond between the cell and the wall actually increases and the cell sticks more. And so if you think as a physicist, a classic physicist, almost none of your intuition works about cell adhesion. We're going to assume that the physics works, and this is a huge oversimplification of the biological reality. You can add the complexity back if you want, but understanding it is not trivial. So how do we model cell adhesion? We're not going to model individual cell adhesion molecules, but what we are going to model instead is the idea that there's some surface of the cell, and there's some density of cell adhesion molecules on the surface. And that means that when the surface of the cells come together, depending on what kind of cell is on either side, they stick. And that means that there's going to be an energy per unit area associated with the contact between two cells. In general, keeping the notation of potential energy, a higher contact energy means less adhesion. If the two cells have a negative contact energy, I have to do work. I have to do put energy in to pull them apart. If the cells have a positive contact energy, pulling them apart gives me energy back. And so you have to remember that higher positive numbers are less adhesive to each other. Bigger negative numbers are more adhesive. And what I'm going to do in my lattice is sum over all of the surface of my cell, and I'm going to add up just the way with surface area I counted. Now I'm going to say that the energy per unit surface depends not only on the area, but on what's on either side of the boundary. And so I'm going to define the contact energy between cell type A and cell type B per unit area, which we call J, and I'll add that up. Now, in reality, it depends on the molecules on either side. And there's also something called contact flex that lets you define the contact adhesion in terms of molecular species. And you could do exactly what we'll do now that way. And Josh and I were playing with it earlier today. But it's a little bit more complicated to do at first. So we'll start with a simpler version. And so here's an example. I've defined two cell types, blue and green. They're fluctuating randomly. They have a target volume. And I've defined it so that the blue cells stick to each other more tightly than blue to green, and the green cells stick to each other more tightly than blue to green. And blue sticks to blue more than green does to green. And what I see is that this system fluctuates, and the initial random configuration rearranges until I get all the blue cells in the middle and the green cells on the edge. And that's called cell sorting. Now we can think about this just the way in the homework that we covered last week, where we looked at the contact energy, the volume energy, and the surface energy for different configuration. If we think about cells, suppose we have two species, light and dark, and fluid medium, it's a green, and we specify per unit area the contact energy between light and light, light and dark, light and medium, dark and dark, dark and medium. I can imagine what configuration of these things has the lowest energy. And that's what it's going to try to do. So suppose I just had two cell types, light and medium. And suppose I mix them like this. So every light cell is surrounded by medium. And I suppose I'm just nearest neighbor. So every light cell has 4 medium contact. So the energy of this pattern will be 4 light medium energies per light cell. So I have 1, 2, 3, 4, 5, 6, 7, 8 light cells, 4 bounds per light cell. So 8 times 4 times J light medium. 32 times light medium is the pattern energy. Suppose I rearrange them into a square with a notch cut out of it. Same number of cells. Now I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 light medium contacts. And 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 light contacts. So now my energy is 12 times light medium plus 10 times light. Which of these patterns has the lower energy? Well, it depends on how light relates to light medium. I could take the difference between these two. I go from 32 light medium to 12 light medium plus 10 light. That is, I've saved myself 20 light medium junctions at the cost of 10 light junctions. That means that if 2 times light medium is bigger than light, the compact configuration wins. If 2 times J light medium is less than 2 J light, the dispersed configuration wins. And so I could go through, and actually in this case you could calculate what's called the surface tension, which is just the difference between those. If the surface tension is positive, the cells get pulled together. If the surface tension is negative, they disperse. And I could do this, for example, with 2 cell types instead of cell medium. And I can ask the question, which one of these has a lower energy? And in this case, there are more possibilities. I'm not going to go through all of the different ones. They'll be in the slide deck if you want. But if I have a checkerboard, for example, here, I have 8 light medium contacts. I have 8 dark medium contacts. And I have 24 light dark contacts. If I split them so that the dark is all on the left and light is all on the right, I still have 8 light medium contacts, 8 dark medium contacts. But now I have 10 light contacts, 10 dark contacts, and only 4 light dark contacts. Again, I could take the difference between these two. The number of light mediums didn't change. The number of dark mediums didn't change, so I can ignore those. And so I have to compare 24 times J light dark to 10 light J light plus 10 J dark plus 4 J light dark. 4 of those cancel, so I have 20 light J light dark versus 10 J light plus 10 J dark. Which one is bigger? If the J light dark is bigger, the right-hand configuration wins. If the J light dark is less, the left-hand configuration wins. If J light dark is bigger than 1/2 the average of J light and J dark, then the right-hand configuration wins. If J light dark is less than the average, the checkerboard wins. And you could do that, we'll do that. See when you get a checkerboard and when you don't. But the point of this kind of argument is that you can think about these things and come up relatively quickly with an expectation of what makes sense. And I strongly recommend for any simulation you do to try to think as hard as possible about what makes sense. Because it's very, very easy to write a computer program that gives you some output that doesn't have a syntax error. If it has a syntax error, the simulation will crash or tell you that there's an error. But if you have a logical error in your code, the code will run and give you something meaningless. And so it's very important to try to develop as much as possible an intuition about what's reasonable. And in the case of cell sorting, you can determine by hand every possible outcome for every possible set of adhesion energies. May be tedious, but you can do it. I drew these pictures as yellow on the left and blue on the right, but I could also put yellow on the outside and blue on the inside. Which one of these wins is going to depend on the contact energy between yellow and medium and blue and medium. Because on the left-hand side, yellow and medium and blue and medium have the same contact area. On the right-hand side, yellow contacts medium, blue doesn't contact medium at all. So if blue medium energy is lower than blue blue, I'll get this one. Otherwise, I'll get the other one. So again, I can come up with all sorts of different configurations for these. In this case, the other issue is, the one I said that, and then I was doing a different example. In this case, there's a question. If you assume that the contact energy with the medium is the same for yellow and blue, in this case, if J light dark is less than J light light, the concentric one wins. You can play with those. Yellow inside versus blue inside, then you have to ask the question which one wins. Again, the more cohesive one likes to go to the middle. And we can play with that. If J light light is less than J dark dark, light goes to the middle. If J dark dark is less than J light light, dark goes to the middle. And so there are all of these different situations that you can get. There's some other ones that we didn't talk about. There could be what's called partial sorting. If dark light contact is very costly, you could get stuck. The lowest energy final configuration may be fully sorted, but getting there, you have to overcome energy barriers. There are walls, energy walls that keep you from doing it.
So it's almost like finding a local minimum in terms of energy.
That's right. And that's exactly what this is. This is, in fact, is exactly a simulated annealing algorithm. What we've done here is determine what the global minimum is by thinking. But starting from a random mixture, we can only rearrange our system locally. And so we can trap in local minima. Because we have fluctuations, if we wait long enough, we'll overcome we'll jump out of those local minima. But if the local minima are deep, it takes a very, very long time. And this happens actually if you mix oil and water. If you mix oil and water and shake it, you'll see tiny oil droplets, and the oil droplets will coalesce, and they'll get bigger and bigger and bigger up to a point. And then the coalescence will effectively stop. What you'll see is that the coalescence slows down as the droplets get bigger. And so unless you wait a very, very long time, you don't get one big oil droplet. You get the oil droplets get bigger and then slow down. And what you'll see is many oil droplets that are dispersed. Much bigger than you started with, but they don't fully coalesce. And so the same thing happens here. You get stuck in what's called partial sorting, is a case where you get stuck in a local minimum of energy. And there are this whole set of varieties of cell dispersal where they prefer medium to each other. That can happen. In metastasis, that's what happened. Inverse cell sorting, where the reds are more cohesive, but they also like the medium more, so they go to the outside. Cell mixing, where the reds like the yellows more than they like each other. Incomplete sorting, we talked about. And so there are all of these possibilities. You can specify the adhesion in Python or XML. Typically, we're going to do it in XML to begin with. It's faster and a little easier. It really is better. We should probably at some point rewrite all of our code to use the Python version where we control the number of molecules. But there are more parameters to think about there. The one disadvantage of doing it this way is that if you have two cell types, it's easy. You actually have two cell types in medium, so you have to specify pairwise every adhesion energy. But if you had, say, 10 cell types, you have a 10 by 10 lattice, so you have to specify almost 100 energies. And so the number of energies you have to specify if you have a lot of cell types goes up pretty fast. But for 2 or 3, it's easy. So we have something called contact energy plugin. Plugin name is contact. We list the types of cells on either side and then give it an energy. And the default is that the energy is just set to 10, all to the same number. And the neighbor order is 1. Again, because it's over a surface, we have to determine how far we go in our adhesion calculation. And generally, you should use 2 or 3 for that number. And again, I have to remind you that the bigger the number, the less you stick, which is a little counterintuitive. So let's do that. Let's make a 2-dimensional blob of cells, 2 types, call them light and dark. Include the contact energy plugin in the neighbor tracker. And then see what happens. You'll have to set a target volume for the cells. And I recommend using a target surface also for the cells. Okay? And this will be an exercise we can do. I'll let you try it on your own, and then I'll try doing it in parallel with you. But is it clear what you want to do? You want to draw a circle of cells, randomly of 2 types, dark and light. You're going to specify all the cells have the same target volume, the target surface. And then you'll create a contact energy plugin. And the contact energy plugin initially will have all of the energies of 10. Run it and see what happens. And then start playing with the energies between the different ones and see if you can make them sort out or mix. Okay? So this is the code that Wizard gave us. Again, we have the lattice. I set the neighbor order to 2. You could have left it at 1 if you wanted. They're not going to make a big difference. I created 2 cell types, dark and light. I gave them both target volumes and target surfaces of 50. I turned on the center of mass neighbor tracker and boundary pixel tracker. We don't need that at the moment. And then the thing that was new was the contact energy, and it defines all of the contact energies of 10. And when I run that, I see the initial random configuration. And the cells are identical, so they're not going to sort. And so now try to find a set of energies that will lead them to sort. And remember what we said. If we want the dark ones to be on the inside, means dark energy has to be lower than dark light energy. So let's try that. If I make dark energy smaller than dark light, now the darks stick together more. Now you can see something's happening. Before, nothing was rearranging. Now you're seeing rearrangement. You see the yellow cells have clustered together and the green cells have clustered together. But you also see that they're not fully sorting. And this is that metastable state that Kali was talking about. And this happens at all of these sorting energies. If I now played with the light-dark contact energy, I could maybe make that happen more easily. If I want to do this, the steering window lets me change this, but it's not very friendly because it'll show you the values, but to see what the cell types are, I have to open things up. And so, for example, if I look at dark light here, dark light is 10. Suppose I made dark light 7. What happens? Notice now the yellows are beginning to come together. It'll sort out more easily. Suppose I made the dark light energy smaller than that. Remember what I said about mixing. If the dark light energy is less, you want to maximize the contact between the two. It's called the checkerboard. Now I'm going to go back to making the dark light energy a little bit bigger. But my green cells are going to get stuck in the middle, so it's going to take some time for that to separate. What if I made the contact energy between the medium and the dark cells very small? What's going to happen? Why don't you try that? If I make the medium dark contact energy 3, what's going to happen? Somebody have a guess? Or try it and see?
Would the dark cells begin to start detaching at all?
Exactly. Very good. So you see, you can get a lot of control. There are only certain kinds of configurations you can get. There are certain things you can't do with denser contact energy. But I can make the cells disperse. I could have the yellow cells stay connected and the green cells disperse. Green and yellow both disperse. I could have yellow go to the middle and green to the outside, green to the outside, yellow to the middle. Have them mix. There are quite a few different things that I could do. So why don't people play just for another minute? Has everybody got the basic simulation running? Does anybody not have the basic simulation running at this point? Does anybody need help getting it working? Believe it or not, that was a couple of published articles in Physical Review back in 1993. It seems pretty trivial in retrospect. That's what got me started on this whole thing, was that simulation. Okay. So one of the things that we were going to do today, and I'm going to show this to you, but we'll save the doing it for homework. Because I want to talk about cell growth and division before we end today. We'll come back and do cell growth and division in more detail next time, but I want to get to it. Is how do you quantify this kind of pattern? And basically the way you have to quantify this pattern is by measuring the amount of contact. Ideally, you could look at the cluster size, but the size of the domains is small, so the cluster size is noisy. But I want to measure how much light contact there is, how much dark contact there is, how much cell medium contact there is. And to do that, there are two ways to do it. There's an iterator that iterates over the boundaries of the cell. It'll return that information. But there's also actually a pre-calculated version of this. And I don't know, Josh, if that's in the Twit code snippets or not. So there is an object called get cell neighbor data list for each cell. And that returns information on the cell neighbors. And that does not return a list. That does not return a list. It actually returns a relatively complex object. But one of the things that I can do with this is I can say common surface area equals cell neighbors dot common surface area with cell types, and then list the types. And so I could use this to say, iterate over a cell, look at a cell and say, how much contact do I have with red cell, blue cell, green? I'll get the color right eventually. Yellow cells. How much contact area do I have with green cells? How much contact area do I have with medium? And so that's something, this slide is something for homework you'll need. We're not going to go into the details of it now. But as a homework exercise, I'll ask you to plot versus time for different energy configurations. What happens to the dark contact, light contact, light contact, light medium contact, and dark medium contact. Okay. And I'm not, again, I'll let you look at the slides on your own. I'll upload them. But you'll see you can play games with sorting and look, for example, here the cells are dispersing. And so the contact between cell and cell is going down, between cell and medium is going up. There are a couple of things to remember about surface area and contacts. Again, surface area is measured by counts of neighbors, and we're on a square lattice. And as we found out, a 3x3 square and a 3x3 square with a hole cut out with 8 pixels have the same surface. And so we're going to have sometimes some counterintuitive effects, especially if the cells are small, where the fact that we're jagged means that the surface area and volume don't behave quite the way we would expect. And in particular with contact energy, you'll see that sometimes you'll see the boundaries of the cells will line up on the lattice. You'll see all your edges will be vertical or horizontal or 45 degrees. And that's, of course, not biologically meaningful. That's because the lattice itself is interfering, is giving us preferred minimal energy directions. And if people have done computer graphics, you know this is a problem. If you have finite screen resolution and you want to draw a line and an angle, it gets jagged. And if you want to do graphics, for example, for printmaking, you have something called dithering, which fuzzes out boundaries to avoid lining up on the lattices. And so you're seeing the fuzziness of the cells. If I zoom in on these cells, you'll see that I've picked an energy where the boundaries are fluctuating quite a bit. And that means I don't line up with the lattice so much. So that can be a bit of an issue in some of these simulations. Also, when I change the interaction range, the number of the boundary pixels per pixel changes. And so that means that the numbers rescale in a relatively complicated way. Okay. So if I want to talk now, I want to talk a little bit about cell division. Cell cycle is pretty complicated, very complicated, in fact. And it's regulated in all sorts of different ways in cells. And cells have a series of signals that they use to decide whether they're going to proliferate, sit there and be quiescent, or die. And if they're going to die, how they're going to die. Some of those signals are things like survival factors that keep the cell alive but not proliferating. Some of them are what are called growth factors, which promote cell growth and division. Some of them are apoptotic factors that promote cell death. And the cell adhesion molecules themselves send signals that are related to proliferation and death. So if a cell has integrins, which it uses to attach to the extracellular matrix, and those integrins are not bound to their target, they send a signal that the cell should die. Why is that? Well, if a cell is looking for the matrix it should stick to and it doesn't find it, it means it's in the wrong place. And if a cell is in the wrong place, something has gone wrong. And very often it means that the cell has become cancerous. And so this is a safety mechanism where cells that are in the wrong place kill themselves because they say it's a little bit like a robot, except robots don't usually do that successfully. I guess there's some science fiction movies where a robot says, I've made a mistake, and that kills itself. Well, cells do this all the time. And so there also are external death signals like tunnel, trail, TNF. Cell-cell adhesion tends to be anti-proliferative. So if a cell attached on all of its sides to other cells of the right kind, then it essentially sends a signal saying the tissue is intact and the cells should survive but not proliferate. If a cell has cell adhesion molecules like cadherins that are on its surface and not attached to its neighbors, then that sends a signal that the tissue has been damaged, that they're missing cells, and that tends to start the cell into cell cycle and to proliferate. And so there's something called contact. This is a phenomenon called contact inhibition, which is when cells are not pushing on their neighbors very much, they proliferate. And when they're completely surrounded by neighbors or under pressure, they stop proliferating. And that contact inhibition can work in a whole variety of ways, but cell adhesion molecules are a big part of it. And so here there's a signal diagram of some of the different things that make cells, help cells decide, am I going to grow? Am I going to stay where I am, or I'm going to die? And the actual decisions cells make are quite complicated. In tumors and cancer, those decisions are not made properly from the point of view of the host. In some degenerative diseases like arthritis or muscular dystrophy or cystic fibrosis, those decisions are made inappropriately in the direction of cell death. But we're going to ignore a lot of that complexity now. We're also going to ignore the details of the cell cycle. In fact, cell cycle consists of multiple phases. Most of the time, the cells sit there in G0, where the cell is quiescent. The cell's happy, it's alive, it's doing its metabolism, it's doing its functions, but it's not growing and proliferating. Something happens to kick the cell out of G0 into S phase, and in S phase, once S phase starts, the cell is determined that it's going to have to go through cell cycle. S phase, it synthesizes additional DNA, it replicates the DNA. Then G2, the cell synthesizes proteins that are needed specifically for cell division. Then there's a fairly short cycle phase called mitosis, where the cell actually does the complicated gymnastics it needs to do to divide, to split the chromosomes and divide. And then after that, telophase, the cells consolidates itself. And then if it's going to stay in cell cycle, the cell will grow and produce more messenger RNAs, and then typically arrest. Or if it's going to be, say, a tumor cell that's going to divide continuously, it will just keep going through this cycle. In our simple models here, we're going to assume that the cells are in cell cycle, unless they're not, and the cells are going to grow, and we're going to neglect, we're going to lump together G1, S, and G2 as continuous growth. Our cells don't have DNA in them, so the replication of DNA versus growth isn't something that we know about. And we're going to assume that mitosis happens instantaneously, that the cell division happens instantaneously. Now, in reality, there are checkpoints where the cell can stop during all of these processes. The cell can divide without growing. Early on in embryogenesis, when the egg is fertilized, the egg doesn't produce a lot of new proteins, but except for DNA, and the egg will divide, produce new chromosomes, divide, produce new chromosomes. So you get a cell initial egg volume of V. After the first division, the volume is V over 2. The second division, the volume is V over 4. Third division, volume is V over 8. So it's possible to have cell division and without cell growth. It's crazy in Drosophila, where the chromosomes replicate 10 times. You get 1,024 chromosomes. There's no cell division. So you go through S phase 10 times with no cell division. You don't go through M. And then the single cell divides into 1,000 cells all at once. So there always are these strange things. In chickens, you get S phase where the chromosomes divide, and you get partial division. So the cells go part of the way through M phase, but not all the way. And so you get partly connected cells. So biology always has counterexamples. That's just part of life, I guess. Now we're going to neglect that. So in our model, we're going to, here's a nice, Josh found a nice picture of cell division going on, and all of that complexity. We're going to ignore that, and in our model, we're going to say the following. The cells are going to grow at a constant rate, for example, and when they get to subtarget volume, they're going to divide. It's not a wonderful model, but it's a place to start. And so if we're going to have cells grow, and we'll come back, we're going to run out of time. So I'll walk you through this, and then we'll go in more detail next time. If we want a cell to grow, we're going to have the individual cells have their own volumes. So we use that volume flex that we did. And we've already seen how to make the cells grow. We add to the target volume. And so we're going to say that each time step, the target volume increases a little bit. And then what we're going to do is we're going to say when the cells get to a predetermined volume, and we have to decide whether we're going to have them divide when the actual volume gets to a target a predetermined volume, when the target volume gets a predetermined volume. I'll do something a little bit different, which we can play with. So again, we're going to run out of time. I'll see if we can come back and do this exercise, but I want to walk through the process first. So we'll have a cell grow and just grow, which we've seen how to do. We'll just start with one cell. And then we're going to have the cell divide. So we'll see the cell grow like that. And then we're going to have the cell divide. Now, cell division is, most of the things we've done we can think of as being continuous in time. Cell division is what's called an event. It happens instantaneously, and it changes the state of the system discontinuously. Because in real biology, the division of cells happens gradually, and it can be partial. Cells can split from each other mostly, but not always, all the way. Computationally, you either have two cell objects or you have one cell object. You can't have 1.5 objects in a computational sense. And so we have to make a decision, what's called a fiat decision, an assertion that at time 1, we have one cell, at time 2, we have two cells. The biology may be more complicated. If you're a true slime mold, not a dictyostelium, but true slime mold, they don't even really have a concept of cells. They just have lots of nuclei floating around in shared cytoplasm. They seem to do just fine that way. So what we're going to have to do to make a cell divide is first we're going to have to say the cell divides. And then one of the cells will be the parent. The other cell we'll call the daughter cell. The first thing we have to do is say which pixels of the parent cell stay with the parent and which pixels of the daughter cells go to the daughter. We saw that movie of cell cleavage. We have to do that. And the second thing we have to do is to decide what the properties of the parent and of the two offspring cells are. At the very least, if the target volume and the target surface area represent the mass and the membrane area of the cell, we probably want to divide the target volume and the target surface area by 2 when the cell divides. Otherwise, we've effectively doubled the amount of material when the cell divides. That's assuming the cell, and if divided by 2 would be if the cell division is symmetric. In most cases that we're going to deal with, cells divide equally, but not always. In embryonic development, early embryonic development, you get a lot of asymmetric division where one cell of the daughter is much bigger than the other. So CompuCell has some features to make this easier, cell division easier. It has one feature that is going to be an annoyance. which is that I mentioned that everything we've done in Python is in a steppable. So if we go back to our Python, we had cell sorting steppable. And this inherits from something called steppable paste pi. Because mitosis, cell division, has somewhat different properties from other things that we're going to do, there is a special mitosis steppable. And so typically when you're going to do cell division, you're going to have two steppables, one ordinary steppable that looks like this, and a second one that will be called mitosis steppable. In principle, you could just use mitosis step and put everything in that. But you can't put cell division into a regular steppable because it doesn't have the code loaded that it needs. Once you use mitosis steppable, though, we'll find that it's very convenient because it has a lot of different built-in features. If you want your cells to divide always in a particular plane, you can specify a plane of orientation. If you want cells to divide along their long axis, you can tell it to do that. If you want cells to divide along your short axis, you can tell it to do that. If you want cells to divide randomly, you can tell them to do that. And so you have a lot of geometric options. All of those occur in various situations in biology. There are also fancier ones that you have to do by hand, but those basic ones occur. The other thing is that there's a routine that allows you to have the daughter cell inherit all of the parent's properties. And it used to be that if you added properties by hand, like dictionary entries, you had to copy them by hand. But now it works. So magically, almost, don't believe in magic, but almost like magic, if I add a dictionary entry like pressure to a cell, and the cell divides, the daughter cells inherit the pressure. The things that I have to do by hand are things like adjust the target volume in the target surface, and I do that before I clone the attributes of the cell. The other thing is that I'm typically going to be doing cell divisions, looking at all the cells for some criterion, and if I'm iterating over all my cells, and I have a cell divide, I've changed my list of cells. And remember, iterating over a list that changes is a no-no. And so what typically you will do is you will iterate over all cells in your system. You'll make a new list, which is the list of cells to divide. And then you will go through that list and do the division. So you don't want to do a cell division inside of a for all cells list loop, because that violates the Python principle that you cannot operate on, you cannot change the elements of a list while you're iterating. And so there are a couple of technical issues that we'll run into that we're going to have to deal with. Okay. So when we do it, we're going to have to decide why the cells are dividing, how they divide, are the daughters the same, how the attributes of the daughters relate, and then once they've divided, do they stop dividing or do they continue dividing? One issue that we're going to find, this is the last thing I'll say, it's all technical, but we'll come back to it, is that steppables are like functions. And you remember in a function, when you define a function, variables are local to the function. I can have a variable named x inside one function definition, variable x inside another function definition. They don't clash. And that means that information that I define inside of a steppable in general isn't available to another steppable. And so if I've got my base steppable, cell sorting steppable here, and I have a separate mitosis steppable, and I want to pass information between them, I have to create a variable that either is global so that it's accessible to both, or and the CompuCell, this is a relatively recent addition to CompuCell, just the way now entries to a cell object are okay because the object is passed, but entries that are created in the steppable aren't. And so CompuCell now has something called steppable shared variables, which is a dictionary where you could put things that are available everywhere in the simulation. And that's new, and you'll find that some of our code doesn't use it because it's new. But that's the best way to get information in and out of multiple steppables. So, and then we're going to play a little bit with this. And I was just going to show you, if you want to create a mitosis steppable, we could use an add steppable function. There's also a box for a mitosis steppable in Wizard. And so next time we'll play with that and we'll come back. But that's important enough that I wanted to walk you through a little bit of that today. So the homework for this week will be primarily on cell sorting. There may be one or two challenge exercises where we'll play with a little bit with mitosis and ask you to read in the manuals about mitosis. And then next week we'll do we'll do cell proliferation. We'll do what happens to the pressure when cells proliferate. And then we'll do contact inhibition of proliferation. And then you'll have most of the things you need to do the projects you've talked about. The things we haven't talked about yet will be chemical fields and secretion and absorption of chemicals, and the tactile response, movement of cells up and down chemical gradients, which is called chemotaxis. And when you've got those, which will take another week or two, you basically have all the pieces you need for doing 90% of the simulations you want to do. There are a couple of other things like creating junctional adhesion, rigid structures, and putting signaling and metabolic networks, so gene regulatory networks inside of cells. So those will be still to do. But the basic things cells do and how they work, we'll have by the end of next week, the beginning of the following week. Okay, any questions about that so far? I know that was a sort of a fly-through of cell division. We didn't get to play with it, but I wanted to talk about it now, and then we'll come back and dive into it in more detail.
James, if you allow me, before we part ways, if anyone is still thinking about their projects, I have a co-infection project that I am currently starting to work on. It's going to be part of my dissertation. And since it's going to be my dissertation, I am highly motivated to produce results. But it would also require consistent communication with me. So if anyone finds themselves thinking about projects and they might find the co-infection area interesting, just feel free to email me and we can set up a meeting and talk about it.
So maybe it's worth saying again, co-infection. So several people have said they're interested in understanding viral infection and immune response. So typically, most of the models that we've done so far have been about when you're infected by one virus, which is bad enough. You get COVID, that's bad enough. But in the reality, when you have the flu, somebody can cough on you with COVID. Or if you have COVID, somebody could cough on you with the flu. Or when you have rhinovirus, somebody could cough on you with the flu. And it's relatively unusual for people to get two viral infections at the same time, but it happens. Or you could get a virus followed by a bacterium, or bacterium followed by a virus.
Yeah, right.
Sometimes,
and for example, with rhinoviruses, it's actually, since rhinoviruses are so common, they cause a common cold. They often occur in their co-infection, influenza and rhinoviruses. Co-infections are actually quite common.
And one of the things that's sort of interesting there is that for a virus to infect you, it has to shut down part of your immune systems. Our immune systems are really good. If our immune system were working 100%, it would shut down most viruses before they got started. Influenza has a particularly nasty way of shutting down the interferon-induced antiviral resistance. And so if you get influenza and then a week later you get another viral infection, it tends to be really bad. Flip way, you get another viral infection, it upregulates interferon, and it protects you from influenza. And so this kind of interaction can be very important. This year, because of social distancing and isolation, there's basically no flu. It's actually remarkable. Influenza rates are down like 98% this year because of isolation. So while isolation hasn't worked very well for COVID, it's actually been sufficient to prevent influenza pretty well. But people were really worried because there's a lot of worry that if you got influenza first, a week before you got COVID, and then got COVID, you could die at rates that were 5 or 10 times higher than the death rate from COVID by itself. This year, because the people are still distancing, it hasn't worked out that way. But next year, when COVID unfortunately is still going to be around because it's evolved around the vaccines that they're using, and people are not going to be isolating forever, we're going to have influenza and COVID. And understanding what that means is pretty important. And so if you're interested in doing viral modeling, Josh is working with Amber Smith, who's a very, very good mathematical biologist, but also doing experiments with mice, trying to understand how these infections either support or interfere with each other. And so, I don't know, I guess Tyler and Devin are the two people who were thinking, maybe, well, you were thinking about cancer modeling. But if one of you is still interested in viral modeling, I would say that this is not a bad topic. And it's almost certainly lead to a publication. Maybe not at the end of the semester, but within a few months after that. So if somebody was interested in working on viruses and viral replication, and you're willing to think about what happens when you have two viruses at the same time, it could be a good project. So, okay. And so contact, if you find that exciting, contact Josh. I'm certainly not saying that people should change their ideas, but it is a very interesting project and a pretty good opportunity, I think. So with that, anything else, Josh? Is that?
Yeah, no, that's it. If anyone wants to talk a little bit more about it, feel free to email me.
Okay, well, thank you, everybody. I know it's late. And if you have any questions, please, the people who have not got a formal contact with somebody to work with, please make sure by the end of the week that you've had a meeting with somebody and we've decided on a project. Kali, I know you and I and TJ should meet about the DICTY simulation. Devin and Tyler, I guess, have ideas, and we've already talked a little bit. We could talk some more, and we'll go from there. Thanks, everybody, and I'll look forward to talking with you the next few days and seeing you next week.